I'm interested in age-related changes or age-related decline in voluntary movement control. And what I'll do today is give you a couple of examples of the type of research that I'm conducting leading towards developing interventions to improve voluntary motor control in the ageing population. So it's well characterised that older adults tend to move more slowly than younger adults. The movements are more jerky and they're more variable. And these changes in movement affect tasks of daily living, like eating, hygiene, many things. So what I'm interested in is understanding what's happening in the brain when we perform very complex movements, and how are these processes in the brain affected by age. So one of the main parts of the brain that's important for the control of our movement is known as the primary motor cortex. And it's this green strip here, and within that part of the brain there is a representation for all of the major body parts. And you can see with this cartoon that is a very large part of our brain devoted to the control of our hands, and that's because we're very skilled with them. Similarly, a large part of the brain devoted to the control of our face, and that's very important for emotion and language. So most of my research uses a non-invasive brain stimulation technique to help us measure activity in this part of the brain. And I think you might have heard about this earlier. It's a technique called transcranial magnetic stimulation. We have a wound coil, you can see in the green, which we have an electric field. This induces a magnetic field that actually passes through the skull and induces current flow in the brain, which is able to activate the brain cells. And if you'll excuse the mess, here's an example of what this technique looks like when performed in the lab. So a pulse comes through this coil. The sound is the exact point at which the stimulus is delivered. And I hope that you can see that if we hold that coil over the part of the brain that controls the hand, we have a twitch. I'll just replay that. We send a pulse, we have a twitch. And what we do in the lab is measure the size of that twitch. So it's called a motor evoked potential, and the size of that motor evoked potential gives us a measure of cortical excitability or brain excitability. What we can also do is deliver this stimulation in the form of two pulses. And what you can notice is that if we give a pulse, one pulse, we get a very large response. If we give a second pulse, which is exactly the same in intensity, 100 milliseconds later, the response is much, much smaller. So this allows us to measure inhibition. There are processes within the brain that are important for stopping movement, and we can measure these with our magnetic stimulation. So these types of inhibitory processes are important for stopping unwanted movements and for controlling the execution of those very fine movements that we can make with the hands, those smooth, accurate and fast movements which are affected by age. So the type of research questions that we ask is, does the effectiveness of these inhibitory circuits change with age? And is the efficacy of these inhibitory circuits associated with our ability for fine motor control? So we use the stimulation protocol that I just described to measure inhibition, and we use tasks like picking up small objects and placing them in holes to measure fine motor control. And the results from one of our studies show, not surprisingly, that the number of pegs someone can place, so the performance, on this particular fine motor control task is larger or better for younger than older adults. That's not surprising. What we also showed, which is quite new, is that the amount of inhibition within that part of the brain controlling the hands is different between younger and older adults. There's more inhibition, those circuits are more effective in a younger adult than an older adult. And what we're particularly interested in understanding is whether there is a relationship between what's happening in the brain and the ability to perform fine movements. And what we show is that those individuals, those older adults who have maintained this strong inhibition, this young-like inhibition, are those who can perform the fine motor control task better. So they have a lot of inhibition and they're very good at a fine motor control task. So this kind of study provides us with some evidence of age-related changes in processes within the motor cortex, and it also gives us some evidence to suggest that the decline in these processes is functionally important. It's related to fine motor control. But we're interested in more than just one part of the brain. We've started to look at networks within the brain, and in particular, we're looking at a part of the brain known as the supplementary motor area. It's shown in pink here. It's important for our bilateral movements, like walking. It's important for stopping inappropriate movements or switching to a new movement if our environment changes. And it's important for timing or coordination. All of these things are really important for balance. What we also know is that these are the supplementary motor area and the primary motor area that I discussed before are directly connected. If we apply stimulation to the supplementary motor area, we actually get a response in the primary motor cortex. So we can use our brain stimulation to measure connectivity. This is the simple brain stimulation I showed before. If we actually have two coils over the head, one that targets the supplementary motor area and one that targets the primary motor area, we get a bigger response. This is evidence that we can measure this connection and that it's excitatory in nature. So the types of research questions we ask are, is this connectivity affected by age, and is it related to movement, to bilateral movements? So we use the complex brain stimulation protocol. We ask people to step around in this quadrant to measure their balance, and we ask people to perform the peg task using both hands. What we find, again not surprisingly with the behaviour, is that younger adults can perform the balance task more quickly than older adults. Younger adults also place more pegs with the bimanual task than older adults. These results just characterise what we know about declines in movement with age. When we look at the results from the brain stimulation, it's clear that in younger adults, we see this facilitation, this strong connectivity, but in older adults, that's gone. So what these findings suggest is that we have strong connectivity between brain regions in younger adults, and that strength in connectivity is lost with age. Again, we're interested in understanding how what we measure from the brain is associated with behaviourally. And what we find is that those individuals who have very strong connectivity perform the balance task most quickly. They perform better on this task. And those individuals who have strong connectivity also perform best on the bimanual task. They place more objects. So what these results show is that if you have strong connectivity within these parts of the brain that are important for movement, you have better movement ability. So what we've shown is that, again, we have some evidence of a decline in brain function, this time in connectivity with age, and that this decline in brain function is functionally meaningful. It's related to an individual's ability to perform complex bilateral movements. My research more broadly is looking at a network approach to understand the neural causes of age-related decline in voluntary movement. And what we're doing in the lab is to look at the effect of cognition on movement, older adults relying more on cognition for movement, and looking at motor learning across the continuum from people who have motor impairments to those who are experts like athletes. We're also looking at methods that can strengthen brain connectivity, a variety of non-invasive brain stimulation protocols combined with physical activity. And all of these things we hope will lead to better interventions to improve voluntary motor control, not only in the ageing population, but in those with injury or neurological disorders as well. Thank you.
I wish all my lecturers had been like you. You made that so clear, and it's really quite complex and very foreign to most of us. So that was very well done.
Thank you.
Do people have any questions or comments? Apart from what can we do to ensure that we maintain our connectivity?
Lifestyle factors have been shown to be associated with good brain function. Those who are physically active have more plastic brains. I'm sure everybody here has heard about neuroplasticity. This is the ability of the brain to change with experience. So those individuals who lead a physically active life maintain this plasticity of the brain, which is very likely to be important for these connections.
Stay active. Questions, comments? Yes.
Okay, so thank you for a nice presentation. So if age-related changes are reversible, how much reversible? Like, for example, we can make elderly people to like young ones, or how much we can change?
It's a good question. A lot of research is using these repetitive non-invasive brain stimulation approaches to induce plasticity, and there's fairly good evidence now that the brain of older adults remains plastic. What we're seeing is that the time course of plastic changes is different between younger and older adults. It takes a bit longer for plasticity to be induced in older adults. So we can use that in that knowledge to develop interventions. But I guess that's the ideal if we could make old brains younger.
Keep working hard. Any other comments? Yes, Leon.
How old are these people? And if they were 100, would they have no connectivity at all?
They are 60 plus. The oldest in this sample was 83. Most of them are in their 60s. Would they have no connectivity at all?
Or is it just some people have a disease, have the strokes?
Sorry, repeat the question.
Or is it, you see, if it's an ageing phenomenon, you'd run out at 100 because there's some people who are very bad at this. So therefore, is it just some people have no connectivity because they've got little strokes underneath the top of their brain?
With this kind of work, we can't answer that question. We don't have any idea really about any non-diagnosed pathophysiologies. What is probably very important is a kind of biological age rather than a chronological age. Some people use markers like grip strength to measure this. But it's possible that maybe some of these measures of connectivity or other neurophysiological measures could allow us to determine a biological age.
Thank you very much.
Thank you.
Very enlightening.